In other waters. So new game. Let's continue, but I haven't started the game yet. I don't know anything about this game other than there's fish and I'll be doing some voice acting. That's all I know. That's legitimately all I know. I haven't even watched the trailer. Taxa. All right. Baikal exoplanet extraction. Transitioning. Fellow Traveler presents. Oh. Firefly? A jump over the age game. Created, designed, and written by Gareth Damian Martin. Music and sound by Amos Roddy. Music's already good. And additional programming by Chris Payne. Why am I reading the credits? Small team. In other waters. So many fireflies. What? There's an error. Hold on. What? What? Rebuilding memory? What happened? Uh-oh. I'm being reconstructed. Am I a robot? Log received. Recovering last known log. Log dated 2189. User and sensorium... AN. Sensorium disabled. AN. There isn't any time left. This is going to be more restrictive than you are used to. But you are resourceful. I know you. I think that's like corruption in the data. I'm sorry, Oki. I can't find you. I can't lose everything we here. I think it says archived here. Maybe I should not try and voice the glitches and should try and like, I don't know. I can't lose everything we archived here. I cannot read whatever that is. User disconnected. Continue with boot? Yeah, let's go. Boot me up. Pilot. Hello? Is someone there? This is Dr. Ellery Vass. I urgently need all systems of this suit activating. I've been locked out, and I'm 20 meters below the surface. I say again, this is Dr. Ellery Vass. Can any operator personas on this suit please respond? Alright, good. Operator, activate all systems. Pilot authorization granted. Power? Power panel activating. Plenty of juice. Okay, utilities available. Let's see. Sampling, sample storage, terminal and retrieval seem to be installed. Oxygen panel is up, and rebreather functioning. I'm off of reserve oxygen now. Lovely. I'm seeing the heading panel come online. Excellent. Okay, good. The dive panel is active. Current reading of 20 meters. Scan and topography readouts coming through. Looking good. You don't seem to know what you're doing. You're not an operator, are you? Then what are you? Why are you installed in this suit? Help me out here. I don't know. I knew there was something strange about this suit when I got inside. It looks decades old. Look, we don't have time for this. So whatever program or system you are, I need you to work with me. You seem to respond to voice commands, so that's a start. We need to head north. I'll explain once we get going. But right now, I'm locked out of half the systems of this suit. Someone, somehow, has rewritten the permissions to only respond to internal messaging. That means you. So I'm going to need you to take control. Let's get moving. Take me north, into the reef. Please? Okay, I will try. Into the reef. Go. Ooh. Southern Gully feature. Steep rock walls rise up towards the surface, either side. The floor is layered with pale sand. Vector clear. Go? I'm figuring it out. Just don't want to break them. Good. Whatever you're doing, it's working. Let's keep heading north. Well, we're kind of going, northwest. No, wait. No, is that west? Never eat soggy wheat bix. Yes, we're going northwest. Harrow Pass. Feature. Boulder fills the passage, blocking out the warm light of Gillies 667 CC's 3 stars. Ooh! You're gonna make you eat this soggy wheat bix? It's a fucking thing I was taught in school on how to remember the frickin' cardinal directions. Going around the circle, it's north for never eat soggy wheat bix. It's an Australian one. I've heard various different ones. I don't know why we had to flavor ours specifically Australian. Never enters Santa's work show. That's a different one. Fair enough. Vector clear. We're there? Okay, let's keep going north. Move chat to top right. Is it not on top right? There's very little I can do to make it reverse direction. Like, I wish I could do that. Yeah, okay, I might make it smaller. Try talking again. There you go. That works. That should do. I almost tried to click out of the stream window by hitting end streaming. Whoops. Because that's where I see your chat. So that's where you're seeing kind of where I'm seeing it. So gully opening feature. Here the passage opens out back into the dappled light of the reef's shallows. Insert clip of Kermit turning on his PC with his toe. Gemma. Yeah, I know what you meant. Where are we going? Just keep scanning, I guess. Ooh. Soft currents. The softly flowing water is filled with glittering particles of organic matter. Signs of rich and thriving life all around. EV. Sounds really... Ooh, what's this? All these dots. Look, in the water. Are those spores? And those stalks to the west. They look like huge fungi. This is... unbelievable. Actual alien life! How could Minae have kept this place a secret? I saw her notes at the base when I landed, and some telltale signs on approach, but here it is. The first extraterrestrial life humanity has ever discovered. Oh, I can't believe it. Bed of stalks, creature. Fungal creature made up of a series of stalks. These stalks are beautiful. I wonder how they feed, how they grow, how they communicate. Series of stalks and plates attached to the rock. Color changes from dark amber to acid yellow. This place, this planet, needs to be studied. Keep scanning the creatures, and I'll keep notes. Once we've gathered enough data, I can name and log each species, no matter what happens here. This is vital work. Noted. We will keep doing that. This is fascinating. Bed of stalks. This creature's external membrane appears to be made of layers of chitin, like the Earth arthropods or fungi. Fair enough. Southern nursery. Protected by ridges from strong northern currents, this could be a safe haven for younger, softer species. Beds of these stalks coat the pale rocks. Are these plants or animals? They are unlike any earth coral or sponge. Woo! They ran away. Wait, were those creatures? They were hidden amongst the stalks. Quickly, try and get a scan. Bed of stalks. I'm seeing a complex pattern of contracting, expanding slits in this stalk's membrane. Mouths or sensory organs? Oh. Agile creature, striped with luminous colors. These flexible, flapping creatures move rapidly through the water with little effort. These are such lively creatures, and so colorful too. I'll start logging my observations. Alright, you do that. Oh, I want to try and catch those. These colonies are producing a shifting hum. Perhaps it is a form of communication between the individuals. There's another one floating around. I want to catch it as it comes by. Come on, little poppet. Come on, there you are. Come here. These creatures appear to maneuver with a series of siphon jets concealed beneath their folded mantles. They are incredibly fast. Let's go over here. Oops. This way. Oh, they're running away. Gotcha. Flitting around the reef, these creatures chase stalk spores with great enthusiasm. Is this their primary source of food? These creatures aggressively pursue certain spores, but ignore others completely. Can they detect differences in mineral content? I'm seeing spores entering and exiting the slits on the surface of the stalk. Something is being exchanged. Among these spore-clogged stalks are piles of small stones. Could a creature have gathered them here, or just the currents? Sample candidate. Okay. These stalks are covered in spores of all sizes. Perhaps we can sample one of the larger ones. I'm in position. You'll have to take the sample, open up the utility. Oh my god. Really, dude? I literally have a thing saying no ads. Fuck off. Asshole. If I end up posting this, I'm gonna intentionally block out with like red stuff. I'm not even gonna let that advertisement go on through into a video. That's so fucking annoying. Okay, one of these buttons has got a sample. That one? Biosample, okay. Ooh. Fungal. Hello, we aren't going anywhere until you take a sample. We got one! Biological samples are vital tools for analyzing and understanding creatures. But this suit should also allow you to deploy any sample back into the ocean. How about you test it out? Hold and drag it over the central compass. A stalk spore containing a complex cocktail of minerals and compounds. Let it out. Yay. Great. That's working as it should. Ooh, they're calling out. Look, those creatures are coming for the spores. Why these ones in particular, I wonder? Perhaps different spores carry different compounds? Each with a specific purpose? Or message? What do you think? Could the spores be a way of communicating between different colonies? I think so. It isn't that strange. Fungi on Earth form networks so exchange. Maybe these stalks do the same. Anyway, sorry, I'm not sure why let's keep moving. Hey, just you keep talking. I'm having fun. I'm wanting to collect this again. I want sample. Mine. What is this? That's our sample thing. Map data. Request retrieve. Okay, I don't know any of that stuff yet. Ooh! Bed of stalks. The colony seems to be broken up into individual patches, and then larger groups over a wide area. I don't know what went down that way. Spore flow. Spores from the main part of the reef are brought into the southern nurseries here by steady current. What is it that dictates reef's growth patterns? Sunlight? The flow of water? Whatever it is, must be absent here. Okay, let's go that way. Ooh. New species logged. Fungal creature, which communicates through sound and spores, and undocking ecosystem seen across the reef. I'm uploading my initial notes. Let's call these reef stalks. We can class... Bugger. I really need to, once that pops up, I need to focus on that. Whatever's here, I can read later. This is timed. So I've learned that now. Good to know. Smooth boulder. This huge rock sits on layers of lichen. Could it have been thrown here during a volcanically active period in the planet's history? I don't know. Could be. What does this do? Oh, it zooms in. Cool. This is very minimalist for a game. I didn't expect it to be this minimalist. Haven't been over here. Stalk patch. These stalks are furred with spores, which appear to have drifted from the northern part of the reef. So this is where we haven't... the one that we didn't get to before. Sampling? Ooh, ooh, ooh. Another fungal seed. Mine. We've got 2 of them now, okay? Let's go back up here. Okay, so here is where we haven't gone yet. To the spore flow. There's a flow of spores coming this way. Hopefully they're not toxic. Hey, can I ask you something? Yeah? I've worked with AI programs before, but you aren't like them. They are predictable, programmatic, dull. Their intelligence allows them to log and process data. That's all. But you are different. I feel like you are alive. Do you understand what I'm saying? I do. Then you are different. I'm glad I'm not just imagining it. But what are you? And what are you doing in this suit? I can only answer yes or no. I'm sorry. You're going to have to be more specific. Northern current. The current running into the reef slows here, but further north, the water whips down the reef with incredible speed. Also, what did I do that made me seem more alive? Woo! More reef stalks. Can I see the data that we sent? Damn. So now that's just what it is. Wide and long with sheer rock walls on either side, this deep rift channels unpredictable currents. Maybe we don't want to go that way yet. Overgrown arch. This arch is covered with hardened stalks. Perhaps the slow currents passing through brought their spores to settle here. Don't know. Ooh, those are bigger ones. Ooh, what are these? Large stalk, static creature, made up of one huge flexible stalk. These huge stalks are incredible. Each one a vertical ecosystem. They are leaking spores into the water as the fruiting bodies attached to their skin bloom. Unclear if swaying is due to a current or is produced by the stalk's own body. I wish they weren't timed. That's the one thing. I wish if I flub a line, I want to be able to say it again. Many of these stalks are leaking particles into the water as they sway. Are these spores or something else? Maybe they're eggs. I kind of want to go over here. Like, I do want to know what's those, but... Oh, this has gone further away. Flecked with spores, this water runs hard and fast. The suit can't push any further north without better propulsions. Vector blocked. Good, okay. So I've got... Oh. Shelled creature. A mobile creature with a large translucent shell and 4 swimming appendages, which is used... These creatures have such beautiful translucent shells. I'll start logging data on them as we find them. 4 swimming appendages, which it uses in a rapid rowing motion. This is going to be so broken up if I try and edit this. Oh my god. Gonna have to try and, like, I don't think I can separate the audio. I think I'm just gonna have to leave it uncut and just have people just go, yep, that's understandable. These larger stalks sway around this basin. They almost resemble branchless trees, bending in a storm. Oh, did you see that? The stalks responded to our movement. They extended as we passed through the arch. Perhaps it was the sound we made when we moved through? We should be careful not to get trapped here. This suit's power capacity is limited. Okay. Don't swim into it. Large stalk. The chitin of the stalk's membrane is very hard, but flexibility is given by a surface of overlapping plates, so it's like armored. Feeding grounds. There is a lot of activity among the rocks ahead. Creatures are grazing and living amongst the stalks, forming a complex ecosystem. Oh, come back, you. Shelled creature. These creatures glide among the reef stalks with great care and precision. They appear to be grazing on them. I'm imagining they're like turtle-ish. They are like swaying back and forth. Oh, vector blocked. Hold on. Oh, because we moved through it, they grew over. That's what they meant. We shouldn't be trapped here. Be careful. Look, over to the southwest. That stalk is totally calcified. That looks like a good candidate. Looks like a good sample candidate. Okay. We'll try and get a sample. The plates of the stalk's surface passing over one another must be producing the distinctive hum that can be heard. These creatures must be related to the stalks on the other stalks on the reef, but seem to have a separate purpose to the lower beds. Calcified stalk, sample candidate. This huge stalk is totally still, its plates hardened into a rocky shell. Can it be revived? Or is this what death looks like here? I don't know. Let's go find out. If we can. This stalk looks dead. Open up the sampling panel and we can extract some of the outer membrane. There's lots of dead materials here, so we should be able to take multiple samples. Okay, let's do it. Fungal. Mine. Okay, so the chitin appears to be hardened with calcium carbonate. That could be useful if we need to boost our power. The suit is able to metabolize organic compounds to produce fuel. Just drag them into the power panel. The suit displays how much oxygen or power each sample can produce. No point wasting important samples. With these, we can keep one sample so we can analyze it back on the base and use the other for power boost. Okay. How do I tell? I see. So I think that shows how much. Ah, yeah, only a little. Yeah. Very little from that. Ooh, maybe. What happens if I deploy these? The stalk spore. I don't know. Ooh. It's safe to assume that the singing of these stalks relates to the communication between colonies seen elsewhere on the reef. New species logged. The sing stalk. I've named these sing stalks for that strange hum they produce. What is its precise function, I wonder? You can see the percents on the bottom left, top, oxygen, bottom. Percents. Where? I can, like the, yeah, I see that's the percent. I don't see numbers, I just see the dials. Large stalks, which, when you have the samples open, oh, so fungal is power and tissue is, oh, okay, interesting. 15% of power, I see. You think they're separate? Oh, okay. Yeah, and you can see those little down there is how much it's gonna fill up. Yeah, so maybe those are different. Yeah, okay. Right side is what gives it... Okay, yeah. Oh, right, fungal, yeah. Oh, okay, I assume that fungal and tissue... Oh, maybe that's saying what it is. Oh, yeah, fungal and seed. Oh, I see, and that's how much... That's power, and that's... Oh, yeah, because that's the symbols there. Oh, yeah. I was thinking that was a line, and it's like fungal, 5%, seed, 0%. Yeah, no, this is different. I see. They're two separate boxes. I got it now. Yeah. So the sing stalk, large stalks which sway producing a distinctive hum or groan, part of the network of stalks which is found across the reef. Ah, come back, you. Come back. Come on, don't make me chase you. I'll chase you. I'm chasing it. I think that's a different one. Where did it go? There's another one around here that I haven't scanned. Or maybe that's the same one. We lost him, girls. There's a way up there and a way over here. Overgrown pass. The space between the rocks here is thick with stalks. The elaborate patterns they trace across the reef are beautiful. I think maybe we should go up first? Not sure. Eeny, meeny, miny, top one. Forest path. The growth gets heavier as we travel north into the Stalk Forest. Something about giving it that name makes it feel safer. More familiar. Vector blocks. Never mind. Good. I like that it's linear because it means that I'm not missing something, because I don't want to miss anything here. Like, as much as I like choice in games and stuff, sometimes I like the linearness because it means that I'm not going to miss anything, because I don't want to miss shit. Ooh, agile creature. The relationship between these creatures and the stalks is unclear, but they camouflage themselves among the amber colonies. So sometimes the dots that that float across that don't have the symbols on it doesn't mean they haven't been scanned before, it just means that they haven't been scanned since moving. Okay. Bear Canyon. The rocks here are clear of stalks and other growths. Is their absence a territorial marker, or just a side effect of the conditions of these waters? Unknown. Territorial makes it seem like there might be another creature. Oh, we've been blocked off. Hopefully there's a way forward. Cavern edge. A large cavern drops down into the reef here. Its entrance is shadowed by the surrounding rocks. Hopefully we're not trapped in it. Oh. Are we fucked? No, we can keep going. The water is still in the yawning entrance to the cavern. There are no signs of stalks or other reef life around the entrance. Descent possible. Ooh, we're going down. We don't want to go back to base and like... Alright, you good? Let's go, I guess. Down. 28 meters. We went 8 meters down. Stalk patches. The patchy sunlight coming through the broken cavern roof is enough to allow the stalks to grow here. If that is what they require. Don't trap us down here. We all know how bad diving accidents can be. Tall stalk. It's among the... Oh. These tall stalks seem to watch over the smaller colonies. I'll start logging notes. Tall bubble-coated stalk which sits among the reef stalk colonies, covered in wide irising pores. Irising? Oh, like the door irises, like the sort of doors in video games. I first heard the term iris in terms of a door in Stargate SG-1. The only reason I know that word. Soft sunlight spills down onto a long, thin stalk, stretching towards the light. A coating of large glassy bubbles glitters on its skin. We're gonna sample this stalk. Don't grab us. Okay, it seems fine so far. We have multiple sample candidates. Ooh, lichen, signal. Yoink. These gaseous bubbles are intriguing. The interior gas is at an elevated pressure. If this membrane ruptured, sac would screech as it depressurized. Is this part of the Stalk communication system we've seen across the reef? Wait, I have an idea. Try deploying one by that blocked tunnel over there. I want to see how the Stalks react. Just drag one over. Let me get this last sample first. Spherical gas-filled membrane. I assume she wanted us to go over there to deploy it. Clogged tunnel. Stalks mesh across the cavern here, hardened into a near-solid wall of tubes and plates. Vector blocked. Okay, so we are trapped down here in a little bit. So let's drop the little sack. Did it do anything? Deploy it away from me. Oh, okay. Shrill sack. Oh! Vector clear. They retracted! These bubbles must be some kind of signal system. I'll call them shrill sacks for that squeak. We can use these to get through the heavy growth ahead. Let's get moving. The cavern curves away in both directions here. It's hard to see the way ahead. Blind corner. Uh-oh. Careful. So I guess you're wondering where we're heading. There's a way station just north of here, on a finger of this reef. I want to find it. My shuttle touched down just hours ago at a floating research base to the south. That's where I found this suit. I'm here looking for Dr. Minae Nomura, chasing a message, all the way from Earth. I must sound mad coming out here to a planet in the tail of Scorpius just to follow a message. Wait, do you know where we are? Don't you? Do you know where we are? No, I don't. But how? We are on Gliese 667cc, a supposedly unremarkable world, if it wasn't for all this life. Somehow the exoplanet extraction course must have passed it by. But how did Minae know to come here? Is that why she contacted me? To verify her discovery? But then, where is she? Look, let's find that way station. Then I can start piecing together what's going on. Alright. I'm gonna get a drink of water, because this is a lot of talking I'm doing. One moment. Okay. Let's get moving. Thin passage. The cavern narrows here, and small eddies of water twirl in the silt. I'm assuming she's reading this, and that's why I'm doing it in the same voice. And I'm assuming it's a she, because they haven't gendered them yet. And I'm a girl, so I'm playing a girl. So, there. Cavern entrance. A current can be felt drifting through the cavern entrance, bringing it with it warm clouds of silt. Ascent possible. Yay, we can go back up! Maybe the way station is here. Oh, and I can see my power slowly draining. Up we go! Back up to 20 meters. I think it's only going down as we move. Tall stalk. Stalks of this kind seem to produce different bubbles depending on the nearby stalks. They are the protectors of the colonies, I assume. Glittering stalk. This thin stalk sits among a bed of other growths, exuding bright bubbles through its pores. Large clearing. I can't help but feel some unseen process is shaping the growth patterns of this reef. What invisible borders am I crossing? Let's go get a sample. Let's go get a sample for study. It's another shrill sack. We might need that to get out somewhere. And we need a couple for study. Let's go over here. See if there's anything... Nope. Rock outcrop. These scattered boulders and steep reefs of the southern reef suggest a violent geological past. Shearing of rock from, like, earthquakes or something. More of shelled creatures in the sing stalks. Unlike other isolated stalk, unlike the other specimens, this large stalk sits alone, away from the lower beds. It is hard to tell if this is the beginning of a colony or its end. Stalk passage. The stalks are more sparse between these rock outcrops. This might be the easiest path north, if I can clear the way. Alright, we can clear the way. We have the shrill stalks. Deep forest. The stalks are so thick here. A sea of amber, waving fingers that must be cleared before we can pass through. Alright, let's deploy a shrill sack. Sweet! Let's go. Tall stalks. Some creatures target these stalks directly, perhaps because they keep them away from the other softer stalks nearby. A delicate set of feeding appendages beneath the creature's soft shell seem to scrape the outer membranes of the stalks away. Occasionally, when the creature scrapes the stalks too aggressively, they retract and let out a defensive signal to nearby colonies. There seems to be some tolerance for the creature scraping by the stalks. They only warn the creatures when they cut too deeply. Navigating these dense growths is like finding your way in a labyrinth, moving from clearing to clearing, with little sense of what's ahead. Glittering stalk. Bubbles like huge pearls glint among the stalks. How do the tall stalks produce these strange screeching sacs? I don't know. The growth patterns of the stalks suggest a complex territorial network. Are we underestimating the complexity of these life forms? Who knows? Let's get a sample of these stalks and try and clear away. We're only getting the little bubbles. We're not getting anything from the actual creature. Better to have too many than not enough. Well, we don't want to, like, fuck up the ecosystem, so not too many. Okay, let's... beep. Signal stalk. She's gonna say something. I'm going to call these signal stalks for the signaling role they play in the stalk ecology. Tall, bubble-producing stalks that protect and monitor stalk colonies. A key part of the stalk network that stretches across the reef. Okay. Oh, hello. Stalk scraper. I'm naming these stalk scrapers, for obvious reasons. You can check my notes back at the base. A large creature with a translucent shell and jointed legs for swimming and feeding grazes on membranes of reef stalks. Oh, it scraped too deep and it had to run away. When will you learn? East and west are stalks as far as the eye can see. They fade off into the silt like the towers of a miniature city. More bubble-producing stalks. Are they sentries designed for early warnings or last-ditch defenses of the colony's core? I don't think we need another sample of the membranes, but you never know, I guess. We're full? Fair enough. Good to know we have a inventory. Upper forest. This isn't a forest, but it carries the atmosphere of one being immersed in interlocking natural processes of life and death. Blocked. I could go that way. What's this way? Spore clusters. Spores from the stalks seem to be massing here in large clumps. This might be a good place for to sample them. Yeah, this is the kind of sample I'm looking for. Something to study. We might have to get rid of some of these. Boop. There's more than one. Stalk spore. Seems like it's just a stalk spore. Beep beep. Beep. Full. Damn it. I'm just freaking beeping at them. They must be getting so annoyed. I shouldn't have collected as many as I did. I didn't need them. Oh, hello. The agile creature. I've noticed smaller spotted variants of these creatures. Another species? Or perhaps sexual dimorphism? These might be the males. Oh, he flew past too quick. Come back. Gotcha. Spore catcher. I'm going to call these spore catchers. They are so eager to carry the stalk's spores. Agile jet-propelled creatures with colourful mantles, found hidden amongst the reef stalk colonies, feeding on their spores. Stalk is starting to become a really heavily used word that's I'm starting to flub just by the fact of saying it so much. Let's go this way, I guess. These two boulders are covered with tiny stalks, a coating of amber fur waving in the currents. But there was also a way this way, wasn't there? Yes, there was. Let's go that way. Beep. Okay. Let's go this way. That's it. We are through. This place is overwhelming. So many new species. This is the central reef. I saw on Minae's maps that the shelf flattens out here. Okay, so maybe we want to go back a bit. That seems like the way out. I want to see what was over that way. Forest edge. The last line of thick stalks before a sandy plain of the central reef. So maybe this was just another way out. A shady outcrop. The water beneath this outcrop feels cool, oceanic. Soft currents scrape sand from the rock face. Ripples of silt bounce sunlight back up through the water, giving it a golden glow. Let's go this way. Yeah, down there is where I was. Just to make sure we've gone to everything. A set of smooth stones sitting together in a pile. Could a creature have gathered these here? Is this a nest? It squeaked and ran away. Okay, so now we can safely move forward knowing we haven't missed anything. Central plain. The center of the reef is flat and sandy. With few rocks to attach to, the stalks struggle to seed these waters. I'm going to take another drink of water and turn my aircon on. One moment! Getting all sweaty and gross. Okay. There it is. I see the way station ahead. Is that it? Way station. A way station set up by Minae Nomura. It's seen better days, but it still functions beneath the coating of stalks. Let's go! Let's find out what's going on. This place is looking a little worse for wear. let me see if I can find an access port. Minae never did take care of her equipment. Some things never change. Sorry, give me a sec. Just trying to... Got it! We should be able to access any data stored here. Just open the terminal and take a look. Terminal available. Ooh, spoofing user. Dr. Minae Nomura. Last accessed. History cleared. Log download? Yes or no? Ooh, we filled our oxygen. The power, I mean. Oh, and it's recharging itself. Interesting. I can't read that. We're downloading some map data. Sync complete. Really? Logs have been cleared. I expected that, given the secrecy Minae seems to be operating under. But the map data mentions something Minae calls the bloom, out across the northern rift. She's been going back and forth to something there, studying it. A unique species, perhaps. Minae, what were you doing here? Why would you keep this discovery from the world? From me? I'm talking to myself again. Or to whatever you are. Sorry. I don't mean... Let's head back to the research base. I need to think. With this way station operational, we can call in the base retrievals drone from the utility panel. This drone will be able to pick us up from any area we've got map data for. Let's head to the base. I've got a lot to figure out. Data released to pilot's console. Connecting. Connection complete. Drone ready. Let's retrieve us. Whee! Baikal, exoplanet extraction, transitioning. Safe mode, restarting, resyncing with Balco OS. Downtime 42 hours. Ooh, sample store. Hey, are you back online? Ellery Vass. Ooh. That is a girl's name by my reckoning. Yes. Pretty sharp work for a biologist, if I do say so myself. Hooking up that strange casing of yours to the base took some work. And the OS here does not play nicely with whatever you are. But you should have access to select subsystems of the stack now. Minae looks to have repurposed some of them, so not everything is functional. Comms are shot, the generator is only partially working, and one of the retrieval drones is missing. But I've booted up the dive bay's mapping systems and sample storage. The lab is also online, but they're also online with analysis tools and an integrated taxonomy for logging creatures. Take a look. I've already logged the creatures we discovered on our last dive. For any logged creatures, I'll also put sample requests in their taxonomy entries. To fill these requests, just find and transfer the samples to the lab, then analyze them. I can use that data to add to the creature study. We need to register these sea species. Head down to the lab level and take a look. I'll also mark sample requests on the dive map. That way we can grab key samples for our studies while we are out in the ocean. And we are going back out soon. While you were offline, I spotted a signal. A suit transponder. You can see its location on the dive map. I want to find it. A suit transponder means a suit, and a suit means... Minae. I'll need your help. That suit takes both of us to pilot it, and it's the only one I've got. Once you are done exploring the base, load into the dive map. We can head out from there. Alright! Ooh. Crew terminal. This is the bay. Sample storage. move all these in. Alright. Comms is damaged. Automatic repair not possible. Offsite assistance needed. The crew terminal. The lab. Medical. Non-active. That's gonna be active in the future, I assume. Generator. Ooh. Generator active. Running at... Ooh, that's the sound of it running. Interesting. Running at 34% efficiency. Tidal turbines active. Core stable. Observation blister. Interesting. Oversight. All privacy protocols revoked. User Ellery Vass. Personal log. Entry 1. 2216. Dr. Ellery Vass. Minae is gone, and she left behind one hell of a mess. This base is falling apart, cannibalized to build way stations out in the ocean. The communication array is sliced away, life support failing. Where did she even commandeer this research base from? There is no way Baikal would have supplied it willingly. After what she did to them? What did she call me out here for? To witness her disappearance? Or did she simply want me to encounter, as she did, the impossible life of this ocean? How could it have gone ignored for hundreds of years of exploration and conquest? There are too many questions to even begin. All I can do is keep an account of what happens here, so that if I can't answer those questions, someone else might. It'll be weeks, maybe months, until the next ship passes close enough to pick up my shuttle transponder. Until then, I'm alone. This place is unbelievably important, our first contact with life beyond Earth. But something else is happening here too, and I need to know what it is. Oh, EV stands for Ellery Vass, so she is reading that. Okay, so I wasn't just making it up. Okay, dive map, taxonomy. Ooh. Ooh. Lab analysis complete. Multiple taxonomy entries updated. We don't need multiple samples, it seems. Okay. What about this? Lab analysis complete. Oh, I don't need to read that, I guess it's the same. It didn't even pop the gas bubble. Huh, cool. Animalia novae. Oh, animals. Myometia. Soft bodies. Spore catcher. Observations recorded by Dr. Ellery Vass. Spore catchers are small, soft-bodied creatures which move quickly through the water by using siphon jets. Their mantles, striped with bright colors that shift as they swim, allow the creatures to modify their movement through the water with precise flapping motions. Watching them navigate the water is hypnotic, their rapid turns and loops showing incredible agility. Flitting through the water like a bat or a bird. Spore catchers use this wide range of movement to pursue the various fungal spores that are produced by the reef's stalks. The spore catchers seem to be highly selective about which spores they wish to consume, suggesting that analysis of the stalk's spores might tell us more about the spore catcher's behavior and diet. Analysis of the unique chemical compounds found in stalk spores, and a wider understanding of how stalks use spores to communicate, suggests a more mutual relationship between the spore catchers and the stalks than initially assumed. If stalks are able to tailor the chemical makeup of their spores, it is possible that the select spores the catchers eat are intended for their consumption and contain compounds and minerals attractive to the species. As spore catchers use their color-shifting properties to hide amongst the stalk colonies, it seems increasingly possible that stalk colonies and spore catchers are mutualistically linked, and catchers serve the needs of their specific stalk patches. What those needs are, however, is not yet clear. Analysis of a spore catcher egg, if found, may help us gain a full picture of their physiology. Anthropelta, jointed shells. Stalk scrapers are large, translucent shelled creatures with distinctive swimming motion, which primarily feed on stalk colonies. Surprisingly nimble, despite their size, stalk scrapers use some of their modified limbs to swim through the water and others to scrape away the chitinous membrane of the stalks. This is tolerated by the stalks to some degree, although they will also deploy shrill sacs to scare away the scrapers. Stalk scrapers seem to be one of the more socially active of the reef creatures, and will call to each other when grazing or when frightened. Each species has a unique cloud pattern on its shell. I wonder how these are formed, and what their significance is within the communities. Analysis of the stalk bark shows that it is chitin-based, as suspected, containing glucose and high levels of polysaccharides. This makes an ideal food source for the scrapers, but not only this, it makes the stalk's membrane a vital substance for lichen scrapers' shell formation. The more stalk membrane consumed, the larger and thicker a scraper's shell will become, which explains the variance in scraper shell size and translucency. Going further, the shells may also be markers of a stalk scraper's importance within the community. A larger shell means a more effective grazer, and therefore a more preferable partner. Further analysis of scraper shells is needed to continue this line of inquiry. The reef stalk. Reef stalks are fungal life forms which take the form of a series of stalks and plates anchored to rocky substrates. Their chitin exterior ranges in colour from dark amber to acid yellow, leading them to resemble oversized lichens. This outer membrane is marked with slits which expand and contract, producing distinctive hum. These slits or pores also allow both the release of spores by each colony and the absorption of other colony spores. I'm using this term for ease, but are these really seeds or something else? Both the hum and the spore exchange seems to be part of a complex communication network between individual stalk patches or colonies. However, like earth fungi, the visible part of these stalks may only be a small part of their overall mass. What networks might connect these colonies beneath the ground? Analysis of stalk spores has revealed a rich cocktail of compounds unique to each spore, some of which are previously unknown to human chemistry. This aspect of the spores and their noticeable exchange between colonies of stalks suggests that these chemical cocktails are part of an information exchange network. However, some of these spores also appear to be true spores, used for the asexual reproduction of stalk colonies, suggesting that the information-carrying spores are adapted or modified reproductive spores. Analysis also suggests that stalk colonies may possess a high level of intelligence and cognition, and that the messaging provided by the spores is nuanced and even subjectively unique to each colony. Patterns across multiple spores from a single colony even suggest a chemical signature possessed by each stalk group. Could these be thought of as stalk names? A chemical signature could be a name. Interesting. Ooh, sketch, that would be cool. Sing stalks are towering fungal stalks that, through swaying, produce a distinctive hum or groan. Part of the stalk ecology found on the central reef, the precise role of sing stalks is particularly obscure. Their singing seems to connect with the sonic communication between stalks, as seen from the stalk's use of shrill sacs as an alarm mechanism. But what they are communicating remains unclear. Notably, sing stalks are often coated in sporing fungal growths, which release spores as the stalks sway through the waters. These spores, released high up in the water column, cover a large area of the reef. There is something tree-like in the structure of these sing stalks, so much so that they give the impression of an underwater fungal forest. Strange, yet familiar. Analysis of the stalk bark sample has given some new insights into the sing stalks. Their entire biology is compressed into the chitin plates that surround each specimen. They are completely hollow. In fact, rather than being a single large stalk, sing stalks are actually many spiraling, intertwined stalks, which weave together to form a pillar-like shell. The scraping of these stalks as they move against one another produces the stalk's harmonic hum, which may not be a form of communication with other colonies as first thought, and instead a kind of internal metronome, there to synchronize the movements of each individual stalk within the weave of stalks. But if the sing stalks are the bark that surrounds them, this brings with it a new question. What do sing stalks contain? Further analysis is needed. This is a lot of talking. I'm glad I have a drink bottle right here, because... woo-hoo! Woo! The signal stalk. What are my thoughts on the game so far? I like it. It's interesting. I really want to see where it goes. Signal stalks are tall, thin, bubble-coated stalks which are often found beside other stalk colonies. Like other calodes, as in calodia, as in fungus. Like other calodes, signal stalks have a chitin exterior, but unlike other species, they feature prominent irising pores that allow them to produce the large bubbles I have named shrill sacs. Through this, signal stalks act as the protectors of the stalk colonies, their shrill sacs breaching when the predators approach, resulting in a warning screech that causes nearby stalks to retract. This makes them a target for creatures which graze on the stalks, and only some colonies are able to maintain a signal stalk protector. But how do these stalks produce their bubbles? And what dictates their growth patterns? Further analysis of their physical products and the structure should help. Shrill sac laboratory analysis shows that they are formed from a coating of mucus, sealed around a pressurized mix of oxygen and carbon dioxide. The oxygen shows that the signal stalks are able to absorb oxygen from their surroundings, perhaps through their large pores? Meanwhile, CO2 must be a result of a fermentation process within the stalk itself. This suggests the presence of a fungal symbiote, similar to yeast within the stalk, and the possible production of ethanol as a byproduct of this process, though a tissue sample will be necessary to confirm this. Shrill sac production, meanwhile, seems to be a precise art. The pressurization, gas mix, and size of each shrill sac can differ from stalk to stalk, meaning that warnings can be directed to particular colonies, and even individual stalks if necessary. There's going to be a lot of reading. So we've got Animalia novi, Fungi novi, Plantae novi, and Bacteriae novi. Alright, I guess it's time to go. Ooh. Signal priority. The base has detected a suit transponder at these coordinates. Could this be Minae? We'll have to head to the East Reef to find out. Obstruction. Minae's notes mention something called the bloom across this rift, but until we have a stronger propulsion, we won't be able to cross it. Sample request. Reef stalks route according to the map data. These caves cut back under the forest. Perhaps there are exposed roots inside. A sing stalk tissue sample. Here, where strong currents batter the stalk forest, might be a good place to find exposed or damaged sing stalks. I saw stalk scrapers here when we crossed the forest. We should search this area for fragments of their shells. Spore catcher egg. Spore catcher movement suggests they nest in the safety of the western stalk forest. Let's investigate. Oh, there's so much. There's so much to do. Where to go first? Oh, I want to learn about these creatures, but I also want to know what happened to Minae. This way. Large smooth pebbles sit in the sand, and beyond I can see the sun's catching the amber terraces of another stalk forest. We could go back and continue to look at these stalks. Let's go back first and properly clear the area. Can we zoom out to a wider map? Nope. If I don't find anything, I guess I can just go back up. Nah. Seems I can't go back. This is a much faster way to go. Yeah, let's go this way. It looks like Minae had to salvage parts of the base to build what she needed to survive. She must have been here for many months before I arrived. Some ruined equipment lies in the shelter of 2 boulders. An early way station site, perhaps? Maybe. Let's check it out. Oh, I guess we can't. Across the open sand, I can see faint shapes of stalk colonies rising up in ordered rows. Stalk Forest. Patterns of stalks stretch back into the distance, pathways and terraces of some unknown design. This must be the western Stalk Forest. Ooh, a sample candidate. At the edge of the strange terraces of this underwater forest, the stalk sits like a sentry, coated with bubbles. I guess this is where we get some bubbles to get further into the Stalk Forest. If we didn't have some already. Don't grab too many. We'll grab all of these, but then we won't grab extras unless we need them. Unlike the southern reef, the stalks here are ordered in curving rows, tightly packed and evenly spaced. I am assuming this is blocked. Ooh. Bloated growth. This growth is bloated and distended, the panels of chitin bulging outwards, and between the seams, something flows within. I've started logging data on these strange inflated stalks. They look ill. Bloated stalk. These stalks bulge out from their bases, forming rounded pulsing growths that look discolored and stretched. Okay. Is there not some... Oh, I guess I should scan. Is this not a sample candidate? Maybe because there's stalks surrounding it? But what if I... Oh. What happened? What do I hear? Is it eating away at my oxygen? What was that? Toxic microbial colony found inside bloated stalks. Ooh, interesting. Stalk pathway. To the west, the stalks spread in thick pathways curving between strangely corroded stones. As they curve south, the stalks meet the rising wall of rock which shelters the southern reef. Let's go further west. Let's get some more samples of these, so that we can get back just in case. These curving avenues of stalks seem to mark a loose perimeter around each bloated growth. Why? Good question. The pulse can be felt running through the shelf from the north, making the sand ripple in small, soft waves. The membrane of those stalks is thin enough to be translucent in areas, stretching to breaking point by whatever is inside. This pulsing growth sits in a strangely shaped clearing. What causes the stalks to grow like this? Something I'm not seeing. When alarmed, these stalks burst open, releasing their toxic contents into the local water. Could they be defending themselves? I don't think so. I think you were right the first time. I think they might be sick. I'm running out of these signals. That's the last one. Let's sample some more. It's delightfully minimalist. Stalk pathway. The bloated stalks seem to be precisely spaced out between the stalks. Are we damaging this colony by disturbing them? I hope not. Bloated stalk. The pulsing, pumping rhythm of these stalks' contractions suggests that some process is happening inside them. Very minimalist, but such a relaxing art style. Yeah, it is very relaxing. That is the word for it. Some vein or root must run through this spot as the sand rumbles with unseen movements. Are all the bloats connected? And it's really well written. It's pumping my imagination muscles, and I like it. This side of the colony loops back towards its center, leading deeper between the stalks. New species. That's enough data to log and classify these inflated stalks. I'm going to call them reef bloats. Large bloated stalks that burst easily, expelling the toxic microbes held inside their bodies. Their movements suggests unseen processes. Line of stalks marks the western edge of the colony. In this direction, a rock wall rises up beyond a sandy shelf. Should I go up or... This is where it's branched out and it's not linear anymore, so I'm not sure where to go. Don't want to miss any areas. To the west stands the final pathway of the stalks before the colony fades out into sand and rocks. This stalk sits atop the corroded husk of a boulder, melted down to a little more than a pebble. And that's the one that I didn't do, I think. Pop, I mean. And I like that the ones that you haven't gone to yet aren't on the order, the one where you can just move it and move around. The western wall rises up towards the shallowest part of the shelf, just meters below the surface of this planet-spanning ocean. Ooh. We can descend. Dark cave. Below the steep wall, a dark crescent-shaped sinkhole cuts into the shelf. Fallen rocks from the wall are strewn across the sand. Ah, so it marks in white the ones we haven't been to yet if you're using this feature. Cool. Let's descend. Let's go down. Down. The walls of the passage twist inward with thick veins of growth. We must be heading beneath the stalks. Bloated roots. Thick bloated roots poke through the rock wall and something pulses within them. This is a rare chance to sample the stalk root network. Let's do it. Ooh. Bloated roots sampled from beneath the Stalk Forest. Hell yeah. That's gonna be fascinating. But can we go down this way? Nope. Apparently not. Cool. Oh no, that's marked white because you can go somewhere. Oh shoot. They've come back up. Ah, good to know. Then they got one more. I hope this takes me to a place where I can harvest some more, or I'm fucked. Yes, there's one. Trying to avoid the exact problem I was afraid of being trapped. Oh, we're starting to run low on power. And that won't restore power. That will. Oh, it almost popped it. Stalk pathway. It seems unlikely that the bloats are simply individually diseased stalks. Their growth pattern suggests a system, a process. With its chalky covering melted away by the bloat, this rock has a dull, metallic sheen, spectrums of light reflecting across its surface. Beyond these stalks, I can see a sheer ridge rising up to the north. Let's go. Northern Ridge. North of here, an angular ridge ascends, cutting the shelf in two. The only way further north is to cross the rift to the... My cadence is weird because my brain is trying to process how to say this. The only way further north is to cross the rift to the east of here. Tucked into the side of this corroded rock, a pile of stones with worn holes sit in the sand. Within a hole, something glitters. Ooh! Ooh. We got an egg. We'll take one for fuel and one for study. Egg sampled from a spore-lined nest. Behind this stalk, layers of thick growth surround a maze of rocks blocking the passage back east. Use it to get back out. That should be plenty. Alright. I don't think there's a way further north or northwest. Let's replenish the one that I just used. I'll only keep like 3 of them. Hopefully I should be able to get back without having to use that egg. Oh, it's still there. Oh. I wonder if there is something down here that I haven't gotten yet. There might be. Oh, there is. The rock behind this growth looks as if it has been melted. Its layers whirl in distorted forms, revealing a metallic core. Sat silently amongst the terraces, this stalk bristles with bubbles, waiting to breach at the first sign of predator. I think I've seen this one. Yeah, that leads over to the path, the underground thing. So let's go down this way. Thin passage. Between the rocks and stalks, a thin, sandy passage leads towards the clearing. small clearing between an avenue of stalks to the west. Their ordered rows dissolve into chaos of the southern forest. Oh. Bye. Clouded shell. Fragments of a shell recently shed glint in the sand like shards of a cloudy glass. What creature left these here? We'll get this sample and then we'll have to go back to the thing to do the thingamajig. We've gotten a couple of samples. What do we get? Clouded shell. A piece of a creature's shell with a cloud pattern. Nice! That was exactly what we wanted. Now we can head back. Hopefully we have enough fuel, so we don't have to use the egg. And it was here that there was a pathway upwards. So I'll go that way next. Give my power back. There we go. Link. Request retrieval. We're gonna study these samples. I know we wanted to go find Minae, but oh, it's just too fascinating. Downtime 5.1 hours. Wonder how long we were down there. Over to the lab. Let's analyze these samples. Let's analyze the shell. Stalker scraper, taxonomy updated. The bloat root. Reef bloat taxonomy updated. Spore catcher taxonomy updated. And the reef bloat updated. Nice. So let's read some more taxonomy. Well, let's read the fungi one first. Reef bloat. Reef bloats are large bloated stalks that can be found at the western border of the reef. Intimidating in their size and distorted shape, they resemble inflated sporing mushrooms, covered in nodules of rampant fungal growth. Their membrane, which is stretched by the bloating process to an almost translucent sheen, is darker than a typical stalk, making the stalks themselves a shade of sickly orange. What role the bloat plays in the reef stalk network is not immediately obvious, but their pulsing, pumping motion suggests that the bloats are either processing or producing the toxic microbes that are contained within their fragile membranes. Without analyzing the microbial colonies themselves, this will be difficult to ascertain. Laboratory analysis of the microbial colonies found within the reef bloats, spark colonies as I've come to call them, show that these microbes are not of a group typically associated with stalk colonies. In fact, spark colonies appear to be made up of highly modified algae with a wide metabolic range, able to consume carbon dioxide, oxygen, metal, and toxic compounds with equal ferocity when supplied with a source of light. When metabolizing these substances, spark colonies produce a regular flow of charged electrons, making them ideal for use within a microbial fuel cell, or MFC. Their suitability for this use is suspicious. Are these really naturally occurring microbes? And why are they only found within reef bloats? Something isn't right here. Finding a roof sample from a reef bloat has brought with it the confirmation that reef bloats are not manufacturing spark colonies, but instead modifying, metabolizing, and processing them. Analysis of the bloat root system has shown the presence of microbes at various stages of modification, including those that have been entirely detoxified and their rampant metabolism neutered. If all reef floats are connected to via this root system, then they may be part of a large-scale cleanup operation. With the aim of eradicating this aggressive microbes, reef floats are not diseased stalks then, but a powerful biological engine which are fueled by their local stalk colonies. The scale of this operation suggests that for the stalks, the spark microbes are a significant threat. Ooh. Oh, cool. Oh, I love the sketches. Oh, that's rad. That's fucking rad. Okay, I need to study these more so I can get a sketch. I really wanna. The spore catcher. Analysis of the spore catcher egg shows a strong correlation between the minerals it contains and the minerals present within the catcher's favored spores. It seems that through the spores, stalks are able to affect the catcher's egg production, therefore controlling the volume, traits, and behavior of the catchers within any area of the reef. In a sense, the stalks have domesticated the catchers, leaving the two intrinsically linked as species. The fungi is keeping pets? But the question of what the catchers provide the stalk remains. Do catchers serve a specific purpose? Consuming unwanted or incorrectly configured spores, for example? Or are they kept for other reasons? Could the catchers be kept by the stalks for their companionship and social use, just as humans keep pets? Do they serve a purpose, or are they pets? We don't know. Oh. Oh, they're the ones with like the siphon flaps and jets. Cool. I can imagine these like undulating as like the little jets propel them along. Oh. Visual and oral sensorium. This is cool. That is cool. Chemical analysis of a stalk scraper shell confirms that not only are they made from stalk membrane materials, their distinctive cloudy coloration comes from the stalks too. Stalk coloration, like most fungi coloration, is reflective of the local water acidity and the medium the fungus grows in. This variance of amber, orange, yellow, and even green pigments is passed directly to the scraper shells when consumed, resulting in beautiful whorls and blobs of color. The scraper's shell is a living record of every stalk colony it is fed on, and therefore a marker of territory and social standing. Scraper shells could be used to help study the entire reef and its shifting ecosystem, each one a chemical map of the local history, readable for those who can learn the significance of its forms. Oh. Leg extension points, cloudy markings, swimming limbs, scraping limbs. Oh. I can imagine them like, isn't it so cool? It is so cool. I can imagine them going in like a wave, like a on both sides, like in like a wave propelling them forward. Ah. This is fucking cool. Alright. I want to go this way. And that way. So that way... Wait, something... We'll see what's that way. There shouldn't be much, but I also want to see what's that way. You'd have been happy with this level of scientific discovery, all that, and an interesting plot? Yeah. We haven't even gotten to more plot. We've just been like, scientific discovery! We're looking at new life and fish and weird ecosystems. Let's try and go further down through the Stalk Forest. It seems like this way was where we might find more samples. Yeah. Okay. So I have one more of these. Oop. So now I can get more of them. Is there another way this way? Because I think it's over here that we're wanting. Is there a way? Let's get more of them first before we move on. I wanna get a sketch of the normal thingamajigs. That's such a great reward for like collecting everything. That makes me want to like 100% complete this because I want to see all the sketches of what these things look like. Because it's like your imagination can do only so much before you're like, okay, what do these things really look like? Ooh, this way. There's a place we haven't been. Spore clouds. The relative calm of this basin means the water is clogged with spores released by larger stalks. The safer and more familiar path. Yeah, we haven't gone this way yet. The feeding grounds. Wait, have we been this way? Yeah, this is where we were before. We got through, yeah, Wait, maybe we can't go this way because we don't have better propulsion. Shit. Damn. Maybe we can't complete the normal stalk ones yet. We've already collected the calcified stalk, but maybe we can use it for fuel. I want to know more. I want to know more, so much more. Oh, bugger. I don't think there's anything that way. Wait, no, is there? Was there a way I couldn't go under the... No, wait, down there was where... I don't think there was a way. I think the only way forward is up that way. Let's go back to the map and... have a look. We didn't manage to get anything. Yeah, that's going up there. We need better propulsion. Bugger. Alright. So now, we can go down this way, up into the area that we didn't look at before. It was here that had the branch, I think. Or was it also down here? No, up here was yeah. This western side of the finger becomes increasingly enclosed by rocky outcrops as it leads towards the shallower water. Oh, maybe this is just another way up. Okay. Calmer waters. The central reef's way station lies just beneath a bubble of calm, but just to the north a massive rift brings strong currents through the rift. Reef, I mean. Ooh. Sandy shelf. Hail sand is unmoving here, but to the north I can see the yawning chasm and strong currents of the wide rift. And that's the one I could have gone on the way station. I see, so it wasn't another pathway. The stalks are noticeably absent from the sides of this northern rift. Why do they leave such a clear buffer zone beside it? Let's go up to the close to the rift before we go that way. Whoops. Hold on. I didn't read that. The passage between these rocks leads out into the howling currents of the northern rift. Northern Rift. The Northern Rift is the largest feature, the largest of the cuts into the central reef. Crossing it without some way of navigating its currents would be suicide. Let's go to the edge and just have a look. Impressive. This split in the shelf is incredibly wide and deep. Those currents would smash us against the rocks if we swim out into the flow. We are going to need better equipment if we want to cross that monster. Well, I'm not going, I'm just looking. Flecks of spores and other organic material whip past in the current, dragged into the dense stalk patches in the west. We're not going that way, I can tell you now. Not yet, at least. Not until we have a better suit or propulsion or something. Let's quickly recharge. Bing. The pale strata of this rock tells an ancient story. Might there be a fossil record within such rocks, waiting to be discovered? Maybe. That would be cool. There are 2 dots of utility that we haven't even... There is no... Ooh. Blooming stalk. This stalk is leaking spores from a wide slit in its side. They are carried across the soft sand by currents coming from the East Reef. Okay. Towering outcrop. This feature rises steeply toward the jagged edge of the reef, splitting the strong currents like the head of an axe. Ooh. Signal stalked. Right, yeah, we've read that already. What's up here before we go down that way? Whoops. Northern Passage. Were these rifts and canyons really carved by the currents? Or are they violent fractures, now smoothed by erosion? I don't know. Or... fuck it, let's go do the sample. Choices! This stalk clings to the edge of the nearby outcrop, away from the loose sand of the central reef. Sampling available. It's more the shrill sacs. Never mind, we'll only take 1. We've got 3 of them. It'll be fine. Damn. Oh well. Split gullies. These gullies lead amongst the tall outcrops of the northern edge, to the currents and open waters beyond. Up or down. This looks like it leads to the northern rift, which I don't think I can cross, so I may as well go up to the point before I can actually go anywhere. And then this, I think, leads forward. In the shadow of these huge outcrops, this path leads towards the rift at the north edge of the reef and its fast-moving waters. From here, the reef descends into the unseen deep. Spores spiral by in the fast-flowing water. What else lies out there? This side of the shelf is sliced by the planet's oceanic currents. Look at those flecks of organic material. They are being cycled back into the reef's ecosystem, drifting from unseen places out in the ocean. It must be part of some vast process. Do you think we'll ever understand the cycles of this ocean? Maybe one day. I hope so too. It's just so intimidating. An entire planet of life and just us to study it. Well, in that case, probably not. If it's just us, I'm assuming eventually we'll get some help. Wait, did we scan when we were up there? I don't think we did. I know there's probably some path out there that we can't get to. Oh, there isn't. Never mind. Never mind, we're fine. Alright, let's see what's down this way. To the split gully. Let's make our way east, to the source of that transponder signal. Yeah, but which way east? Here the gully opens... Oh, I don't know what to expect. If the transponder is Minae's... Shit, There's some history between us, but I haven't seen her in years. Not since... Look, you don't need to worry about it. Let's just find her. What's happened between you two? Friends, lovers, family, colleagues, who knows? Here the gully opens out onto one of the rifts, cutting deep into the bedrock of the reef. There, look, a cave entrance, cut into the side of the rift. It should allow us to bypass the rift, although I'm not sure where we will merge. Who knows? Rift edge. The sheer walls of the rift plunge into the dark water here, and rapid currents make passing through difficult, if not impossible. Hidden shelf. Protected by sheer sides of the rift, this shelf leads down into a small cavern entrance in the reef wall. Let's see what was up there first. Because that seems the way forward. I want to see everything. The walls have been smoothed into soft ripples, the sign of a strong current descending from the north. Yep, so that's this current. Ooh. Scream! These stalks block the gully, but also the strong currents behind. Even if we clear them, those currents will still block our passage. Fuck. Bugger. Well, at least we know. We need better propulsion. Same for there. Cool. Let's descend into the hidden shelf. We've gone all possible ways as far as I know. Down. 8 meters down. Curving passage. The round mouth of the cave stretches away into the rock. Stalks hum quietly in the dark. Moody. There's a strange evenness to the walls of this cave, almost as if it was a vast borehole drilled into the reef. The stalks even seem to get down here. Perhaps they are less reliant on sunlight than I initially thought. I don't know. We are deep in the rock now. What formed these hollowed out caverns with their strange, undulating walls? I wonder how the stalks survive without sunlight. Maybe they are fed by the forest above? What do you think? Perhaps. It would make sense. A network of colonies tunneling through the entire shelf? I don't know. Protecting a small underground colony, this tall stalk's signature coating of bubbles glints in the dark. Let's grab some stalks. Just one more. 3 should be enough. Exposed roots. A cave-in has severed some of the sections of the stalk's root network, stretching down from the forest above. What can they tell us? Yes, what can they tell us? This is exactly what I was looking for. Ooh. Stalk root, a tangle of silken roots from a stalk colony, carbon enriched. Hell yeah. Wonder what we'll learn from that. Maybe we'll even get a sketch. The cave starts to get tight in here, and in the shadows ahead, stalks crowd the tunnel. Ooh. The stalks here are thick and intertwined. Underground, in the stark light of the headlamps, they resemble brittle, oversized worms. Bright light from the east of this large cavern is broken up by tall pillars, casting strips of light and dark across the smooth walls. So moody down in this cave. These pillars punctuate the cavern, stalks crowded around their bases. They support the vast weight of the reef above. These stalks are filling the water with spores. We should start logging information on them. These domed stalks are pumping out clouds of short-lived spores. They look like oversized mushrooms. These bloated creatures are pumping out clouds of spores. Clouds of spores into the water. Around them, stalk colonies rise out of the stand. Are they like the grandmothers or something? These spores seem different. The stalks nearby seem to be stimulated by them. Clump of spores which seem to stimulate stalk growth. Could these spores be used to make stalks extend? To bring them out of hiding? We should test them out on a retracted colony. I hope I didn't miss one message there. Oh. Does work. Unlike other stalk spores, which survive until they settle, the spores from these stalks dissolve moments after they are released. These stalks seem to be shriveling up. Is it because they are too exposed to the rift's withering currents? Don't know. These sporing stalks seem to require very particular conditions to grow. I've seen so few elsewhere on the reef. This long stalk sits quietly beside a dying colony. Soon there won't be anything left for it to protect from scavengers. Another rift. How can we cross it? The current here is so strong. Currents slice down through the rift from the north here. Crossing is going to be difficult unless the flow can be slowed further up. These large growths seem to be sustaining this colony with their spores. Perhaps they carry nutrients? Even chemical signals? Let's find out. Soothe spore. We don't need the stalk bark, let's use that for power. The far side of the cavern narrows into darkness. I can feel the faint pulse of a distant current ahead. The current must have once run down this tunnel, scraping the walls into smooth lines. Now the stalks block the flow. Well, are we going to be blown away if we... One way to find out. No, we seem to be fine. The tunnel ahead is thick with stalks, scraping the ceiling with their rounded tips. I misread it as lips for a second there. The light filters in from a large opening, making the spores drifting in the water glitter like sparks. Tucked behind this pillar, perhaps this stalk grew here as an early warning for predators coming through the cave mouth. Weird cadence there, but alright. Look, those stalks in the cave mouth, they are retracted. Perhaps if we could make them extend, they would block the current. Yes. Bonk. There we go. The cave mouth ahead leaves this passage exposed to the full force of the northern current. Blocking it somehow will help us pass. They block the current! These spores encourage them to extend and fill the tunnel. With these soothe spores, we can control the stalks to our advantage. This will be useful. Pulsing growth. Another of these growths support a smaller colony at the cavern's edge. Is there a logic to where these grow a pattern? Who knows? Maybe we will soon enough. There's a definite correlation between struggling stalk colonies and these sporing stalks. Are they helping support these colonies? Tucked into the rift wall, this shelf allows protection from the fast-flowing water cutting through the rock into the heart of the reef. Okay. Look, out in the rift, a retracted stalk colony. If we can get them to extend out, they'll provide a shelter from the current. Yes. A handful of stalks cling to the rift wall here, blown into spiraling shapes by the rushing currents. Extend! And the current's blocked. Splattered with spores, the wall here feels the full force of the current. Perhaps it can be slowed upstream somehow. Which is exactly what we did, as long as they don't go away. Even this close to the wall, the darkness of the deep rift is intimidating. A huge slit in the face of this world. How deep does it go? If the current can be slowed or navigated, this is the place to do it, where the rift narrows and a gap opens up on both sides. Yes! A huge pillar sits out from the wall here. Its top is caked with retracted stalks, a golden garden waiting to emerge, which we have just made emerge. And we've crossed the rift. This small opening in the wall provides a little shelter from the howling rift, allowing some stalks to take up root here. We are across! Thank you, stalks! Oh, and thank you to you too. You're welcome. Another smooth tunnel leads out towards the light. Gonna take another drink of water, because I'm dry mouth. I wonder how far away we are from the transponder. A circle of warm light shows this tunnel opens out into the upper shelf of the East Reef. What lies above? Up we go! We're about half power right now. Ooh! Blooming petals. These plant-like creatures seem unrelated to the reef stalks, their soft petals waving on thin stems. These are beautiful plants. Single petals shifting in the sunlight. Let's start logging data on them. These bright wide petals appear to be photosynthetic plants. They float on long stems, shifting and swaying in the water. It feels good to be out in the sunlight again. Let's head northeast along this finger of the reef. The transponder is at the tip. Wait a second. There was another one of the things over here. We're not going down, we're just having a look. These canopies are spectacular. I'll start logging observations. Let's see what we can find out about them. These stalks are twisted together into a huge canopy. Incredible! They are pulsing as the pores across their surface open and close. Oh, so they're different. Ooh, we can sample. Ooh. Getting petals. We're running out of space here. While each plant seems to have a single wide petal, they group together forming fields of blooms, all angled toward the sun, like sunflowers. Still sands. The fractured outcrops all around break up the worst of the northern current, protecting this sandy shelf. The coloration of these canopies is much lighter than the other stalks. They glow pale yellow in the light of Gliese 667 cc's, 3 suns. The petals seem to shimmer as they twist and open. It's difficult to tell if this is involuntary or part of the plant's own movement. The colony of stalks has twisted itself tightly across this gap in the rocks. Could they be protecting something? What are you protecting? Where the rock shelf has fallen away into the rift, these roots of the canopy above have been exposed. We could sample them here. Tell me your secrets. We're learning. There's a way station up here. Lovely, I was starting to get full. Broken gully. The outcrops on this shelf are cut through with huge cracks, as if they were somehow shattered. What could have done this? Stalks have grown across this gully, reconnecting the two huge pieces of rock that look like they must have been once one. It's our last... We won't go that way yet. We don't know if the last one... If there's any... I don't know if there's any more. This towering canopy of woven stalks pulsates in the soft current, the sunlight slipping through the gaps in its branches. On some petals, there is a thick clump of pollen on the lower part. I wonder how this is dispersed. It seems too heavy to float away. High up in the wide canopy, I can see that the stalks are twisted around strange ribbed growths. What are they holding up? Shimmer blooms. I'm going to name these plants shimmer blooms for the way they catch the sunlight. I wish I could share these discoveries with Minae. There's still so much to say. Wide single-petal plants that come in a variety of hues. They appear to be primarily photosynthetic and produce a heavy, furry-looking pollen. Bright petals line this part of the reef, shimmering and swaying, parting easily as I pass among their delicate forms. Ooh, sampling is available. We've already got the bright pollen, and we're full, so we'll forego that for now. Yes! With their strong resemblance to terrestrial mushrooms, I'm gonna call these reef caps. Fair enough. Domed mushroom-like stalks which appear to support struggling stalk colonies with supply of special spores. This canopy has many gaps between its stalks, as if something has fallen out or been removed from it. Fed by spores, shielded by a colony of stalks, this part of the reef seems to have fostered the ideal conditions for its own survival. Is that another way station ahead? Minae must have built them all across the reef. In the center of this shelf is a beautiful clearing, screened from currents and predators. No wonder Minae set up a way station here. Okay, so the way to the way station is clear. I can go back. Ooh. Table stalk. I've named these table stalks after the table corals they remind me of. I'll log my notes back at the base. Towering canopies of stalks, intertwined around ribbed growths, they pulse as their pores systematically open and close. Right, so we're gonna quickly go back, seeing as we don't need this last shrilling thing. Beep. Broken boulder. Stress marks can still be seen along the edge of this huge rock, but it remains silent about its own history. Yeah, rocks don't talk, I'm afraid, lady. The stalk looked silently out over the deep rift. I wonder if Minae looked over this same view with different eyes. I don't know. Slashed stalk. This bubble-coated stalk has large gashes in its side and seems to be shriveled away. Its exposed tissue would be easy to sample. Yes! Yes, 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 yes. Mine. 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 Strong current. Across this wide rift, its tall rock walls of the reef's second finger can be seen. What lies inside them, I wonder? Can't go that way. And we don't need more shrill sacks, because we're going back to the way station. So full of research! Oh, we're gonna have so much to read. So much lore. Yep, yep, yep. In the scattered shadows of golden canopies and knee-deep in shimmering petals, this way station sits silent. Hell yes. Okay, open up the terminal. See what data we can scrape from this place. Spoofing user, Dr. Minae Nomura. Last accessed. History cleared. Log download? Yes. After we grab this. Map data! The log suggests Minae was using this as some kind of staging ground for expeditions over the drop-off. She was looking for something out in the ocean. Something deep from the descents she logged here. Minae doesn't do things at random. The way station's the research base. This is calculated. I feel like we are already 10 steps behind her, and we've barely even started. Let's go to that suit transponder. It isn't far from here. If we head straight north to the shelf's edge, it should be there. Ah, but you see, first, we gotta deposit our samples. What if we don't have any inventory? We don't have any inventory space to pick up whatever's we find over there. And you gotta sleep. Never mind. 3.6 hours? How long have you been awake, woman? Stalk segment? Segment of the bubble producing stalk. Canopy root, a root of a stalk canopy. Many twisted filaments. Bright pollen, furred pollen extracted from petals from the East Reef. Soothe spores, clumps of spores which simulate stalk growth recovery. A tangle of silken roots from a stalk colony, carbon enriched. And we still have some of this for some fuel. Alright. Let's analyze. Updated the signal stalk. Updated the table stalk. Updated shimmer blooms. The reef cap updated. And the reef stalk updated. Fuck yeah. Tell me your secrets. Yes! Close analysis of stalk root networks reveals something surprising. The root networks are in fact part of a symbiote, a different fungal species entirely, which exists within stalk bodies. This fungal symbiote provides a carbon exchange network which runs throughout the bedrock of the reef, connecting all colonies in a single web. This symbiote also produces the spores which the stalks use to communicate, but these spores are edited by the stalks themselves to act as reproductive agents, information carriers, and chemical delivery systems. If the stalks are able to edit the genetic signatures of these spores and their parent species, their intelligence and complexity must be way beyond what I previously theorized. Close analysis of the stalk spore language could reveal an entire corpus of communication. Are there stalk jokes, stalk histories, or even stalk literatures? Cool. What about the signal stalks? Chemical analysis of the signal stalk tissue samples has confirmed the presence of a yeast-like fungus, and large amounts of ethanol saturated the tissue itself. This prompts a rethink of why many creatures target the hard-to-get signal stalks as foodstuffs. While I initially theorized it was due to their role as protectors of the stalk colonies, the significant presence of ethanol in the signal stalk tissue means most creatures would feel the effects of the alcohol consumed, and may get buzzed from grazing on the signal stalks. This, in moderation, would be favorable, even enjoyable effect for the creatures, leading them to repeatedly target the signal stalks. If I remain stranded on Gliese 667cc for long enough, I may need to make use of this particular biological property myself. She may have to drink the frickin' reef alcohol. Hell yeah. Cool. The table stalk. Pale yellow canopies formed from many stalks entwined together which reach impressive heights. Table stalks are part of the reef's many interlocking species of stalks, though these individuals can only be found in the eastern reef. There they tower over the other creatures, providing dappled shade for the life that grows around them. One of the notable features of the table stalks is its rhythmic opening and closing of their pores, which provide a steady flow of water beneath their canopies. This seems to be highly beneficial to the growth of the other species, with most table stalks being surrounded by many types of stalks, and other species entirely. Also notable are the ball-shaped growths of differing sizes which sit in the table stalk canopies. Sampling these might help us understand the table stalks better. Analysis of a table stalk root shows a number of minerals not associated with stalk growth being extracted from the bedrock. Do the table stalks need these minerals to survive, or are they using them for some other purpose? A cross-section of the roots shows a central chamber typical of the carbon transfer system seen in other stalks. But unlike those stalks, table stalks have many subsidiary chambers, like bundles of wires, each carrying a particular cocktail of mineral compounds. These secondary roots are not drawing up large amounts of these materials, just traces. But why? Perhaps there is some connection to these growths in the upper canopies of the table stalks. They are well secured by the stalk's tight binding, but perhaps we can find one that has been dislodged. The reef cap. Large domed stalks, like terrestrial mushrooms, release spores from gills on the undersides of the cap. In the wider stalk ecology, reef caps appear to play the role of emergency response, growing in areas where the stalk colonies are failing or are heavily predated upon, and proceeding to shower them with spores. However, these spores seem to differ from those released by other species of stalk, in that they quickly dissolve when expelled. Analyzing these spores is important to better understanding the reef cap's role. Are they reproductive spores, tasked with adding newly seeded stalks to existing colonies? Or nutrient spores, intended to boost the health of dying or diseased individuals? Soothe spores, the name by which I have come to call these reef cap spores, don't seem to be spores at all. Unlike the modified spores of other stalks, these particles don't have any reproductive capacity. Instead, they resemble endospores, dormant bacteria reduced to dried husks, which can survive for hundreds of years in toxic environments without nutrients. Endospores are incredibly tough and high in calcium, and when released by the reef caps, they dissolve and transfer this calcium to the surrounding environment, where it is absorbed by the local stalks. The windfall of calcium granted by the cap's soothe spores seems to allow the stalks to augment their internal signaling and toughen their membrane to protect them from predators, allowing them to recover rapidly. But the question remains, how do the reef caps produce these endospores? Tissue sample required. Punctifolia. Shimmer blooms. Shimmer blooms are single-petaled plants found primarily in the sheltered waters of the East Reef. As they produce pollen, as well as photosynthesize, I have classified the visible portion of these plants as petals, though they could also be thought of as leaves, serving both functions simultaneously. Shimmer blooms can be green, like most terrestrial plants. However, they can also be found in yellow, red, or purple in color. The image of a patch of blooms is a beautiful one. As they shift in the soft currents, they often seem to shimmer. The lower part of their single petal features many stamens, which produce fluffy, clumped pollen. Analysis of this pollen may provide some insight into their life cycle. Laboratory analysis of shimmer bloom pollen reveals it to be heavy, sticky substance, atypical of waterborne distribution. Unlike on terrestrial plants like seagrass, whose pollen is distributed by water currents and is therefore long and light, shimmer bloom's spherical, heavy pollen seems intended to resist distribution by the current, and must instead be distributed by creatures who visit the blooms. The reason for this may be the shimmer bloom's tendency to grow on exposed shelves or pillars, which would risk lightweight, water-transported pollen being sucked into the deep ocean, rather than being distributed to viable sites. By relying on creature transport, bloom pollen has a better chance of reaching another sunlit shelf or pillar. Cool. Now. Ooh. Reef cap tissue. Notice tiny piece of reef cap drifting down this long rift. If we can fight the current, perhaps we can find the individual shedding them. Where strong currents batter the stalk forest might be a good place to find exposed or damaged sing stalks. We'd have to get in the rift. Reef's middle finger. On the reef's middle finger, where the forests and currents fight for dominance, perhaps we can find a table stalk willing to give up its secrets. Whoops, not yet. Shimmer blooms, shoot. These pillars tucked behind the fingers of the reef are only just being germinated by the shimmer blooms. But can we sample a young shoot here? Alright, let's go down. Down. Let's find this story. Like many of the large rocks on this shelf, this outcrop has been split, creating a passage to the east and another to the north. So up here is the transponder. Surrounded by shimmering petals, a pulsing stalk pulses out spores. Do these carbon-rich particles feed plants too? Dunno. Nah, it's just soothe spores. We should sample it because we don't have any soothe spores. Eastern Passage. As the shattered edge of the reef curves away to the east, the currents fade and static water grows warmer. From the cover of this canopy, I can see a series of tall pillars out to the east, each one topped with a garden of unknown life. A strong current cuts across the east edge of the shelf, with the turquoise haze of the open ocean stretching beyond. Damn. Back we go! A swirl of sand suggests this passage leads to the reef edge and the open water beyond. This side of Fingers feels exposed, but thanks to its current splitting the outcrops, it remains in a bubble of calm. Exposed clearing. Is Minae gonna be here? Are we gonna find her body? Or just an empty suit? At this tip of the shelf, the outcrop falls away to reveal an unrestricted view of the vast ocean. It is both terrifying and beautiful. I see something at the edge. Is that a suit? Minae's suit. Open, empty, and half sunk in the sand. Why would she leave it here? And more importantly, where did she go? It's definitely Minae's suit, but no Minae. Surely she isn't. Okay, let's not jump to conclusions. I want to take a look at this. It can't have been here long. Most of it's still in good condition. But how did it get out here? There's no evidence of injury. A malfunction, perhaps? What do you think? No. No, you are right. Then she left it on purpose. Abandoned it. Did she intend for us to find it? Minae, what did you get yourself into here? It's better equipped than our suit, but the seals have started decaying. What's this? Minae has rigged up some kind of custom propulsion system, probably to deal with the currents. We can use this. We'll need a power source. It won't run off the suit system. It seems like she fashioned some kind of microbial fuel cell. We'll need to recreate it if we want to use it. Okay, let's get this back to base so I can take a better look. Minae, you better not have done something stupid. I didn't come all the way out here to bury you. Let's go. Call a drone for pickup. Okay. Also, hi, Doom Player's biggest fan. Welcome. We're playing In Other Waters. We are an AI, unknown AI in an unknown suit of someone trying to find... a scientist trying to find their friend. Back with me? Yeah. Good to hear. Let me update you. I've spent the past few hours working on Minae's suit. Bad news is, the majority of it is unusable. And apart from that propulsion system Minae pulled together, but we are going to need to find a power source. Minae's suit logs mentions her building a fuel cell using microbes from the Western Reef. But they don't say much else. She's been incredibly secretive while she was here, covering her tracks across the reef as if there was someone watching. Or she's just... I don't know. The Minae I knew was tricky, complicated, but not paranoid. Either way, if we want to recreate a propulsion system, we'll need those microbes. When you're ready, let's head to the Western Stalk Forest and see what we can find. Alright. Personal log 2, Dr. Ellery Vass. Minae's suit opens at the ocean. Empty. What has she done? I'm trying to reconcile the Minae I once knew with the evidence in front of me. When I met her on Kepler 62F, she was intense and quiet, but calculated, intelligent. She wasn't someone to take unnecessary risks and leap into the unknown. At first, I found her impossibly intimidating. Her experience working as a biologist far outstripped my own. Michael brought me to the site as a consultant to examine her discoveries, but I felt like the one under examination. The base was much like this one, a smaller, older model, sunk in a subglacial ocean. Someone might think a relationship was inevitable, given those intimate conditions. But it wasn't the base that brought me to her. It was that intensity. Over the few years since, I've tried not to dwell on what happened on Kepler 62F. But being here, living where she lived, following her scattered steps, it's impossible not to remember. Were they... What happened between them? Did they actually get into a relationship, or was it a one-sided thing? Did she love her but not love back, get loved back? Was there a relationship, but like she was too engrossed in her work and pushed her away? I don't know. I want to know what happened between these two. I hope they're gay. I hope to God they're gay. But I'm biased, obviously. Minae's notes on her microbial fuel cell design mentions sourcing 4 microbe colonies from this part of the reef. Alright. We can't go anywhere else over here because of the currents, I'm pretty sure. Maybe we can go back once we've gotten the propulsion system. Let's go! How long have I been going anyway? Ooh, almost 3 hours. We might stop after we've gotten the propulsion system. Because there's only so much I can talk and voice act before I start to get tired. I want to be able to give my full performance to this game, because I'm so interested. Minae's map data mentions a microbe which only occurs in this Stalk Forest. If we can find 4 healthy colonies, it will make it the ideal basis for the propulsion system's fuel cell. Alright. I think we need to get some of the bubble stuff from here. Maybe I should have left the samples back in the base. I don't need this many. I'm already full inventory. I don't need this many. All of these are studied already. Oh, are they talking about the microbes? What are these strange inflated stalks? We know these already. They look like sporing growths we found in the East Caves, but there's something wrong with them. We need to break them open somehow, so we can sample the microbial colony inside. Let's see if they respond to the shrill stacks. We've already done this. I think we did it out of order a little. We've got one, but the clouds of microbes these stalks release are incredibly toxic. The suit is having to flush its vents with oxygen to protect me. We've lost a lot of reserve oxygen. We're going to need to be fast when sampling these bloats. Let's get to another 3 colonies. But be careful, I don't want to run out of oxygen out here. Noted. We can sample here. We'll be fine. We'll be fine. Beep. Ah, bugger. Ah, damn it. Well, thought I needed 2. We might only need 1. Nope. I don't think we can get cultures from here. Okay, let's use the canopy route. There's something different about the growth patterns in this forest. The ones further south were more chaotic, but these stalks are almost arranged avenues or terraces. Do you think they might be protecting something? Perhaps. But what could they be fortifying? And why all these diseased bloats? They're not diseased, we found this. They are factories. Great, now we have plenty of these microbial colonies. But where did this infection come from? The stalks seem to be struggling to deal with it. Or is this just part of their expected life cycle? What do you think? Neither. I agree. This feels like a rare event. An epidemic, perhaps? But what is the source? Either way, let's bring these back to the base as soon as we can. We can analyze the samples here. There. Call in a pickup. Do we have enough? Oh, we do have 1 back at the base, so yeah, we should be able to... She told me to call in a pickup, so let's go. We know they're factories. But where did this bacteria come from? Very intelligent of these plant life. Hey, you ready to get back to work? Yeah. Great, because I've had a very productive few hours. I've been working on that propulsion system. I've used these 4 microbial samples to grow a new colony in the lab. Then I transferred it to a secure containment capsule. Nice and snug. I've followed what I could from Minae's notes on building a microbial fuel cell. It's all loaded onto the suit. The MFC fed directly from our reserve oxygen supply. Now we just need to take the suit out for a dive and test it. Going to take plenty of oxygen to fuel the colony, so bring any samples that'll help out. But make sure not to take any that we haven't... Make sure not to take any we haven't put through an analysis first on the lab level. I don't want to burn through hours of research material just to get a power boost. This propulsion system will let us pass through strong currents and move more quickly. We should be able to cut across that rift, north of the way station, and finally see what the bloom is. Ready when you are. These are all for fuel. We don't really need fuel, we need oxygen. Ooh, the shrill sack will give some oxygen. Bloat root will do a lot. I kind of don't want to get rid of something that we don't have copy of. Okay, that should be plenty, in theory. Yeah, that should be plenty. But I've read this one already. Yep. Alright. Nothing new in here. Wait, we've researched this stuff already. Yeah, all this stuff is researched already. Hmm? Oh! Oh no, we've read this already. Yeah, we've seen this already. We don't need to worry. Yeah, all of this is already researched. Cool, 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 cool. I know I said we were gonna finish, but I want to know what the bloom is. I gotta know. I gotta know what the next step is. We don't have any other objectives other than research, other than the bloom. I gotta know. Gotta know. Okay, so you'll find the controls in the utilities, where sampling and everything is. Once you've found the propulsion system, let's try it out. Remember, it uses oxygen. Be careful. When you've got the hang of it, let's try to cross the Northern Rift. Oh, that's activating and using it. I see. Okay. So that's doing it and that's using it. Okay. Let's go! Broom! This massive split in the ocean seems almost too violent to be natural. Both sides are almost impossible to see from the center point. This pillar rising up from the ocean floor deep below provides shelter from the rift. There's something on the other side. A huge green cloud rising up from the shelf. That must be the bloom. Less dramatically shattered than the central reef, the northern shelf slopes down gently through the layers of sediment. The green wall of a vast bloom of some kind rises up ahead, swirling and obscure. Ooh. The bloom. What ecological crisis spawned this green mass? How is it connected to the reef? If the answers are anywhere, they are inside. Uh-oh. The water inside is totally anoxic. The suit's rebreather can't function. We'll have to run a reserve while we're inside this place. The bulbous and shiny, these formations sit under a coating of green filaments and growths. The water here is both highly oxygenated and temporarily filtered of its toxic microbes by the fan. There's a fan? There's life, still hanging on inside this toxic bloom, creating safe zones in order to thrive. Perhaps these creatures can help us learn how to exist in these toxic conditions. Let's take some samples to see what effects they have on the bloom when we deploy them. Alright. Ooh, and they provide oxygen too. These pale, delicate fans are almost bone white against the clouded green of the bloom. These fans seem to be reoxygenating the water. I'll start logging notes. This should be useful. Okay. Fan dust. What happens if we deploy it? Ooh! Let's replenish some oxygen. The fan sits among shards of rock, trembling in cyclical currents of the bloom. Ah, well that didn't do what I thought it would. It didn't last as long as I thought it would. I should have read before I deployed it. pale fan. Occasionally this fan shivers, releasing a flurry of green growths that have gathered amongst its gossamer spines. Here, the rocky shelf of the reef flakes away into large, fragile sheets with razor-sharp edges. Bivalve creature. Huh? There's some kind of shelled creature hiding in the slit over there. We need to figure out how to tempt it outside of its burrow. Perhaps a sample from elsewhere in the bloom? Something that reacts with the microbial growth itself. I don't know. Maybe the sooth spores? Unsure. A rise of silver silt fills the depression created by the corroded rock shelf. The tip of a shell pokes above it. Is that an eye? Away from the rock outcrops and fans, the visibility in the bloom falls to near zero. The shattered rock catches microbial strings drifting in the water, draping themselves in a shifting web of hair. Well, it's out of its burrow. Nothing but sharp rocks and green water. Those fans seem to grow close to the rocks, perhaps shelter for some of the stronger currents. The plume is vast. What could cause and sustain these processes on such a scale? With the oxygen gone, the microbes here must be metabolizing some other substance in order to persist. These fans have to be filtering the water of the bloom to feed. Behind each one is a small area of clear, oxygenated water. These creatures bury themselves to hide from the bloom's toxicity, making them one of the few life forms that can survive the bloom. In the rusted holes of the rock, silt has gathered, forming a rare shelter for life in the bloom. The bloom is blindingly thick here, though to the southwest something glows between the microbial clouds. Metal traces. A faint signature of a metal alloy, perhaps the remains of equipment Minae brought to the bloom. What was she doing here? What was she doing here? There. Huh. Layers of rock stick out from beneath the silt, eaten away by microbes of the bloom. Alright, let's head down to the metallic thing. The rear side of this fan free of the green strings of microbial growth has a pale ivory sheen. There is a faint fizzing that emerges from these fans. It appears to be caused by their spines reacting with the bloom. Like stone tumors, these rounded rocks rise up out of the green toxic clouds. That makes me feel better. And we've already been there. Cool, just making sure I've mapped out the place. The burning clouds of the bloom roll across these rocks. The microbial life of the bloom blankets every surface. What other life can survive this toxic landscape? Through the green clouds, something bright glows in the dark ahead. Up ahead, a fizzing membrane of oxygenated water. Is this what allows the species here to survive this constant toxicity? Tucked into the ridge wall, this bubble is filled with oxygenated water, filtered through some unknown microbial process. To the south, the central ridge of the bloom reaches up, disappearing into the green clouds of the growth. Strange, glittering domes marked with ornate patterns of holes sit in the silt. Whatever inhabited them is long dead. Woo. Splitting the bloom, this vast ridge descends to the north, where it may be possible to cross. Protected by the sheer wall of the bloom's central ridge, these open nests flutter with the movement of their swarms. Nesting creatures. These swarms of bright creatures provide safe zones in the bloom. Let's start logging data on them. Within these complex transparent structures, the blurred activity of many individual creatures can be seen. This must be their nests. Don't mind if I do. Animal. These colonies do well to hold out against the bloom. The individual creatures seem to be able to digest the microbial growth. We can use this. We should try deploying a colony to see if we can create our own safe zones inside this toxic flood. Oh, nest workers. These strange forms scatter around inside the bubble. Are these skeletons? They resemble radiolarians, but far bigger in scale. They are incredibly ornate. What's radiolarians? A colony of shattered domes lying in the silvery silt. What were these creatures? I don't know. I must say, I am fascinated. Let's go. Blackened rocks. The rock here is mottled with dark patches as if burned by electrical currents. Toxic waters. Traveling this deep in the bloom is like swimming through the interior of a vast green dome. So it's still creating a safe haven in here. Rocks here blend into world shapes. What natural process caused this? I don't know. To the north, the bloom whips in cycles of heavy growth. Oh, damn it. Let's just go. Let's just go. The thin streak of oxygenated water behind the fan shimmers faintly with traces of catalytic dust. Bloom fan. I'm naming these bloom fans. When we finally get out of this place, I'll update the base's taxonomy records. Pale, delicate fans that feed on the bloom by filtering it through their bodies, using a catalytic dust to counteract its toxicity. Fan-like fins which can fold down, and a simple set of retractable eyes enable the creature to totally conceal itself within its shell, like an armadillo. Silt has gathered around the base of this wide outcrop, and signatures of life forms buried within it can be detected. Toxic waters. Among these green, I can see tiny blue flashes, occasionally flickering. Eaten away by the bloom, this outcrop is marked with patina of tiny holes. This way, I guess? Okay, we've been this way. Anchored to the flaking rock, this fan sways softly, dislodging clumps of green growths as it does. The currents of the bloom grow stronger as I move away from the rocky outcrops of its southern side. Let's grab some more. I know there's one still down here that I haven't been to, I can't forget that. A low silt drift provides just enough cover for a bivalve shell, which peeks out from beneath the pale wave of sand. When active, these creatures feed directly on the microbial growths of the bloom, rapidly clearing a patch of oxygenated water. Yeah, I noticed. Green clouds drift above the rock floor, honeycombed with holes. Retracted glow casts shattering... Yep. Fuck. I'm trying to not die from oxygen loss. A fractured glow casts the shattered ground in a cool light. Within the protective barrier, the water is oxygenated and clear of microbial clouds. What chemical process is being enacted here? Like the nests themselves, which are woven from transparent threads, the bubbles which protect them also seem to have a delicate skin. Within the open structure of these nests, swarms of glinting particles form shifting shapes. The ornate domes and porous spheres of the skeletons glint with the purity of their silica growths. So now I can feel like I can use these more freely because I have more samples. And I can use one of these skeletons because they have a lot of oxygen in them apparently. Sphere fragment. Oh, there we go. A small construction drone from the base, corroded beyond use. What was Minae building out here? Beyond the bubble, the green clouds gather again, with water sapped of all oxygen. What can be fueling this endless growth? Oh, I could have done both of them. In this northern part of the bloom, there is little shelter from the green storm. Green in every direction. I can see nothing. There is little to observe this far in the bloom. This fizzing border between the bloom and this oxygenated water within is a welcome sight. Just going to it first, ask questions later. The nest is the way station's... The way station? Protector. Without its swarms of iridescent life, the station would be little more than a rust-eaten frame. There's a way station here? From here, the bloom rolls out over a vast plain, scraped clean of sand by vicious currents. This is as far north as we will go. Yep. When their protective bubbles begin to fail, these creatures burst from their nests to repair them, swarming out to weave more threads. Cool. These beautiful, lifeless shapes, like sculptors' abandoned works, gather around the way station, rocking gently in the current. Starting to run out of power, actually. Okay. I'm not going to the way station yet. I want to explore these bits of it that I haven't found yet. Like down here. The nearby bubble sits alone in this flat plain, scorched by the chemicals processes of billions of metabolizing cells. This side of the bubble seems so close to the way station. If it were to shrink, any foothold we would have in this green desert would be lost. It must have been a struggle to assemble the metallic cage of this way station so deep in the bloom. What led Minae out here? Don't know. Ghosts of some equipment lie in the silt, one of many pieces lost in Minae's expedition of the bloom. A large bulbous rock emerges out of the rock floor. Silt gathers in the winding cracks that score its surface. Bivalve bloom vein. These strange little creatures are getting a big name. Bivalve bloom veins. What do you think? Yeah. I like it too. We can log them into the taxonomy back at the base. The rock outcrops of this area are clogged with strings of microbial growth which dance in the light current. Yes! Huge plates of layered rock make the floor here into a jagged pattern of small overhangs. Blue flashes flicker across the shattered strata of the rock. Partially concealed among layers of rock, this fan slowly undulates, releasing a catalytic dust in its wake. Shards of rock with green clouds between turn the seafloor into a miniature landscape. Sickly pale green light filters down onto the rocks. Down or up? Oh shit. There's one of the things there, I can make a safe zone. Between these two rocks, a pile of shell fragments has gathered. A bronze one glimmers among the green. We could sample it here. You don't need to drop it. It's already opened up. Alright. Yes! That was what I was looking for. Between these sharp rocks, a pale strip of sand leads on through the green. Jagged edge splits open in the rock itself, place for the green clouds to gather. Corroded shelf. I think we've been here. We have. So then down here is the other area I didn't go to. Wait, have we looped around? Is there no other way? Maybe there wasn't. I think that's it. Rot juts out at strange green angles. Oh. Okay. Fair enough. Ah. Bugger. Well, let's try and get back over to that, Gotta be careful. We should go back to the station. As much as I want to keep exploring. Finally, a way station. With this active, we can call for retrieval all across the bloom. Useful when we run low on oxygen. Open the terminal and we can resupply, and see if Minae left any map data behind. Lovely. It's incomplete. Okay, I've got our logs here. Lots of trips back and forth between here and a laboratory. That means she must have set up a remote lab somewhere deeper into the bloom. She spent months making trips to it, ferrying some materials from elsewhere in the bloom to there. She was studying something. Something important to her. We have to find that lab. I'll log the coordinates on the dive map when we get back to base. Alright. Back to base. Only 3.6 hours. How long is it taking between swimming? When did she last sleep? Hey, are you back with me? I am. Good. I wanted to ask you about the bloom. I've been trying to understand what is causing it. It seems unlikely that we are looking at a stable ecosystem. It has to be a unique event. And yet, creatures have rapidly and effectively adapted to these conditions. How is that possible? There's something here we aren't seeing. Something Minae was investigating as well. Do you think she knows something we don't? I think so. She has to. Maybe she's holed up in the lab mentioned in the map data waiting for us. Let's find that lab. Bivalve bloom vein. Bloom veins conceal themselves in bronze-colored shells most of the time, only coming out when the opportunity to feed presents itself. Highly attuned to the currents and flows of the bloom, bivalve bloom veins take advantage of any break in the bloom's flow to unfold from their shells and feed. They primarily feed on the bloom's microbial growth itself, able to quickly establish and maintain a feeding zone of oxygenated water. When a bloom vein tires, it will retreat back into its shell, burying itself in silt once more. I would like to understand more about how the bloom veins are able to feed directly on the microbes of the bloom. Considering the extremely high toxicity, perhaps something we uncover here could be useful for future expeditions into the unforgiving landscape of the bloom. The bloom fan. Bloom fans are delicate and pale fan-shaped creatures that filter the toxic microbes of the bloom. As tall as a human, with a wide span of pale spines that form a feather-like shape, they are instantly recognizable in the green-cladded water of the bloom. Anchoring themselves into the sand with a central stem, the bloom fan's filtration leads to a small patch of oxygenated water on the leeward side of the fan. Bloom fans seem to be sensitive to currents, adjusting their angle for better catchment of the microbial growth, and also can often be seen shivering in order to free their delicate filaments of bloom growth. How can they contend with the high toxicity and deoxygenated waters of the bloom is unclear, but perhaps analysis of the dust that coats their spines may offer some clues. Analysis. Zip. Wait, I can't analyze the sphere fragment? Huh. Oh, I haven't... Right. I haven't seen enough of them. Yeah, I can't analyze them because I don't have a taxonomy entry. After being lucky enough to come across an entire bloom vein shell, analysis has provided some possible explanations for the creature's neurotoxin resistance. The shell itself was found to have a radial pattern of tiny holes in certain areas, all of which were found to have high concentrations of bloom neurotoxin. It seems that when the bloom vein is in its hidden resting state, it exudes the neurotoxin through its shell, processing the glucose in the bloom's growth separately. This exuded neurotoxin coats the bloom vein's shell and the surrounding area, making the creature potentially deadly to any predator. This adaptation to the bloom is therefore a highly effective defense mechanism, even if the bloom vein itself is constantly on the edge of a frenzied neurotoxin-induced death. A close analysis of the fan dust shows that the fans catalyze the bloom's microbes into massively increasing their oxygen production, to the point that the cells themselves are torn apart by the oxygen that bubbles out from within them. This is why the bloom fans can be heard to be fizzing when in close proximity, and suggests that the oxygen is the primary nutrient that the fans require from filtering. Interestingly, this analysis suggests that the bloom's microbes are oxygen producing by nature, and may be a form of algae. It is only their sheer density blocking their access to sunlight, which means they no longer produce oxygen, but instead choke and poison the area. While it seems unlikely that the fans live on oxygen alone, what other nutrient sources might bloom fans use to support themselves? Well, up there is the bloom fan, and up there is bloom froth. And what was down here? Anyway, I think that might be a good time to call it. Either we can investigate some of these areas over here, or we can go further into the bloom. Wait, data from the way station places Minae's remote lab in this southern part of the bloom. She's hiding something here. That's very close. Wait, no, it's not. Oh, that's all the way up here. So going down this way would... I see. So it's pretty far. It's pretty far. I hope that it's saving automatically. If it's saved anywhere, it probably saved when I just got in here now. Ah, save and quit to menu. Message log. Oh, so if I missed something, I can go into the message log and read it. Oh, there's shortcuts as well. I don't have to click. Cool. That's a cool game. I can't wait to see more. Thank you for watching. See you later.